Ladies and gentlemen, dear colleagues, it is my pleasure to welcome Professor Javidiho from the University of Connecticut. He has visited our OSA Charles University in Prague chapter and prepared a traveling lecture about 3D imaging for displays, visualization and computation imaging. His research is focused on 3D microscopy, optical imaging and biophotonics.
Okay, well, thank you very much. It's a pleasure to be here. I want to thank you folks for being here and the OSA chapter for organizing my visit. It's my first time to Prague, and I should say that I'm really impressed how beautiful the city is and how nice the people are. With that said, my research involves with general 3D imaging, and we sort of have a philosophy in our lab. And our philosophy is that, of course, we can't do everything in 3D imaging, but we would like to be a lab that's focused on 3D imaging and to the best of our ability to do excellent research in both sciences and applications for 3D imaging, 3D visualization, and so on. So for now, of course, 3D displays are hot, as we say. There's a lot of industries interested in that, so we get funding in this area, which is very helpful because we can support students and support our lab and so on, and also the area of 3D visualization that has many applications in medicine, in industry, of course in the United States, your research, if you want to get funding, there should always be a component that is related to defense, whether you like it or not. So there are a lot of applications for defense. And what I would like to say, computational imaging, but this is different from what computer science people call computational imaging. To me, this is a combination of both optics and computers, algorithms, and so on, to enhance the viewing experience and benefits. Now, we are told to say a little bit about ourselves. We are in Connecticut, University of Connecticut. Connecticut is kind of sandwiched between Boston and New York. So if you're going from New York City to Boston or vice versa, you go right through our state. There's no other way around it. It is apparently, I didn't know, one of the wealthiest states in our union. Not that I'm one of the wealthiest people in the United States, but per capita income is very high, and that's because there are many companies. You probably recognize General Electric, United Technologies, Xerox. There's also various banks and so on. So it's very beneficial to our university in the sense that we get a lot of funding from the state and these companies. I hope someday you visit my university and you see the benefits of sponsorship. They want us to talk about ranking of our university. We are now ranked number 19 among all the public universities. By the way, I really don't believe in this ranking. You probably don't have anything like that in your country. We have a lot of universities, so people have to see, and universities are expensive, so people have to pay a lot of money to go to college. So this has become very important for universities because students look at this ranking to decide which university they want to go to. If they're going to spend, I don't know, $30,000, $40,000, $50,000 a year to go to college, they want to know what the ranking is. So our ranking is very respectable as a public university, although I don't really believe in this ranking. I believe in this ranking. This is a ranking by National Research Council. This looks at, measures the output of a particular program and a department in terms of how many papers you publish, how many students you graduate, what is the citation of your faculty, how well-known your faculty are. We did extremely well. If you look at this, we were ranked number 33 overall. Overall, there's probably 150, 200 programs. And these are some well-known universities, Johns Hopkins, Carnegie Mellon, and so on. We are in that category. So overall, we were 33. As a public, we were number 17. Okay. These are some quick facts. We're about 30,000 students. This is our operating budget. I had a student from one of these small countries. I will not mention the name of that country. And he jokingly told me that the budget of our university is bigger than the budget of his country. So that's because we are a large university. And with that said, I'll skip this. This is just some shots I thought you might be interested to see. We are very well known for the autumn colors. We apparently have one of the most vivid colors in autumn in the world. If you visit us in this time of the year or in October, you drive around or walk around, you see a lot of different colors, red, yellow, and various shades in between for the colors of autumn. These are some pictures of our campus. We have a campus that's also on the water. We are not located on the water, and that's not campus on the water. Okay. So with that said, we get to the technical side of things here. These are some of the research that I'm doing. Is not all, but some. And of course, 3D displays are there, 3D visualization, computational imaging, 3D imaging with very few photons. This is something we started, which is very interesting. And we can show to you that with very few photons, we can do very complex tasks. We evolved to require billions of photons to be able to operate. We are functional during the day. We go to sleep at night. So as a result, the sensors we have and the algorithms we have were done with that in mind. And we have shown that, in fact, you can, with few photons, you can do some really exciting and interesting things. And when you have few photons, you're working with far less information than you do otherwise. And that makes the process more interesting, but at the same time, you're storing a lot less information, you're communicating less information, you can have faster electronics and so on. This is another field, which is 3D imaging, quantum imaging, and authentication. And likewise, if you can encode your information to be functional with very few photons, and use that for security and encryption, attacking that is much more difficult because the attacker is working with a very sparse data, and they have to figure out what your original information is. This is work we have done 3D imaging in scattering medium, including turbid water, and that's the work on using 3D imaging for automatic identification of cells and bacteria and microorganisms. And the idea here is that you simply present your sample to the system, and the system can decide for you, classify that in an automated fashion, tell you what it is, whether it's what kind of bacteria it is, what kind of cell it is. Is it a healthy cell? Is it a disease cell? Is it a cancerous cell? We are working with one of my former students in India. And what we have done, we have designed a very compact, I will show you if there's time, system that they can take to the field in these remote villages and look for malaria. Malaria is a big problem in India, kills a lot of people. And then another problem is HIV. And these villages have no access to advanced care, so this could be very interesting to apply there. Now, binocular vision is the ability to maintain focus on an object with both eyes, creating a single visual image. No one knows how humans do this, but we do it, okay? And the other interesting thing is that there is constant movement and motion here, and because of that, even though there is a blind spot where the retina is interfaces with these nerves, we can avoid that due to this. So human brain can do this in some miraculous way, and this is basic principle. In fact, Da Vinci noticed binocular vision, and he, in his Trattato della pittura parte quinta, he said, in fact, that shadow light and perspective, any light that falls on the bodies below equal angle, held the first degree of clarity, and there are darker corners that receive less light. And in fact, Da Vinci also has another drawing where he shows that light coming from a 3D object is radiating spherically in different directions. So the early timers knew something about perspective and 3D. They just did not have the resources, the math, or the instruments to make precise measurements. But this is probably the most common type of 3D display, is a stereo display. It's very simple. You have left eye, you have right eye. The idea was invented in mid-19th century. And it's simple. You can display large 3D images, high resolution, has a very large depth, but has problems. And the problems are that certain portion of the population, including myself, when they're exposed to stereo, they encounter what we call visual fatigue and abnormal effects. If you watch stereo, you can get, some of us can get headache, you can get nauseous. And the reason for that is that with stereo, you have the left and right images displayed on a screen like this. And the viewer is wearing glasses, or there's some kind of a viewing instrument or something like that. And the 3D is formed away from the screen, somewhere here, depending on the depth. And if it's a long depth, of course, you have a large depth displaying the object. Now, when your eyes see the photons coming from the screen, you focus on the screen because the photons stimulate the brain. It's out of your control. Even if you don't want to, this happens. You're focusing on the screen. But the 3D image is formed away from the screen. So now your eyes focus on the 3D image that's away from the screen. And what's created is called convergence accommodation conflict, because your eyes keep going from the screen to the 3D object, away from the screen. And this back and forth creates problems for your brain because we didn't evolve to see 3D objects that way. So ideally, what you like to have, and the other problem, of course, these glasses are they're very inconvenient. If it hadn't been for these problems, you would have had 3D TV, 3D, everything 3D. Everything that you see now on 2D would have been 3D. So ideally, also, and you have the problem of parallax here. You don't have full parallax here. So ideally, what you like to have, you want to have a continuous viewpoint, full parallax, no viewing devices, and no visual or abnormal effects. Now, this is the ideal 3D, hypothetical 3D image and display device. You have a 3D surface. These are the optical rays. The optical rays, the capturing system has to record two things. One is the intensity. Photography does that for you. When you take a photograph of a scene, it captures the intensity of the rays, but not the direction. And also, you need the direction of the rays. If you have these two, you can simply propagate these back and reconstruct the 3D surface. Now, photography can't do that for you. Why? Because while you have the intensity of these rays, you don't know the direction. And as a result, everything looks flat. Everything looks 2D. Holography does that for you in a different way. Holography does that through interference of what we call an object beam and a reference beam. And by doing so, you're recording the optical path here that is coming from the 3D surface. And therefore, you can re-illuminate this hologram with a reference beam and reconstruct this 3D surface. Of course, the problem with holography is that coherent laser, which is used, generates speckle. And also, you really have to be careful with the phase uniformity, because if you were to display this on a special light modulator and reconstruct the 3D surface, you require phase flatness on that special light modulator. Difficult to produce 3D color images and difficult to get holograms for outdoor objects, because you would have to illuminate an outdoor scene with a laser beam. And if I were to ask you if I could make a hologram of you in this room, of course, people would be uncomfortable to be illuminated to lasers. So those are the problems with holography. Now, what we have been doing, we have been really integrating what we call computational optical sensing and 3D imaging for 3D display here. Here you're using computational resources to obtain optical and physical information images. Now, and this is done through what we call integral imaging. And a lot of inspiration of that comes from nature. This is the fly eye structure, where you have a lot of small, tiny lenses rather than one big lens. And flies can do some interesting tasks with this. So the idea, maybe I go to this diagram, is that if you have a 3D object, remember I said we need the intensity of the rays and the direction of the rays. So one way of operating with incoherent light or ambient light is to have a sensor device. This could be either display or sensor, but in the recording stage is a sensor. So you have a camera or an array of cameras, an array of lenses, and each camera is recording a different perspective of this 3D object. So it's as if you are getting 2D images, but you're going around the object and getting many, many different perspectives. Now, folks, what is that got to do for you? Well, because you are recording a 2D image, you are getting the intensity of the rays, okay? But because you're sampling the optical rays with these different lenses, you're also getting the direction of the rays. So you're accomplishing what I said earlier about reconstructing a 3D surface. You're getting the intensity and the direction of the rays. And as a result, if you take those 2D images, display them now on something like a large liquid crystal display device, and project them back through an identical array of lenses, what happens is that you are retracing these rays back and you're reconstructing the 3D surface. Now, as you can see that when you project these back, these rays intersect on this surface of the object if they came originally from that point. Okay, so think of a point source here. If you have a point source here on the surface of the object, this gets projected to a point source here, right? So you get many, many different perspectives of that point source. When you reconstruct, you project this back, the rays will intersect exactly where that point source was. Anywhere else, they're out of focus. So if you come to my lab, and if I were to do this for you, unlike stereo, the 3D is reconstructed by the intersection of these rays on the surface of the object. So whatever the surface of the object is, you see bright spots, and that's where the intersection of the rays are. So this remedies, this does not have the problem that stereo has because your eyes are not perceiving this 3D that's reconstructed away from the screen. The 3D, in fact, is generated by intersection of the rays, and you get a nice, bright array of point sources on the surface of the 3D object. It was originally suggested by Lippmann, this idea of using array of lenses for the capture of light, and I believe that was 1908. Yeah, 1908 he won the Nobel Prize for a method of reproducing colors photographed based on the phenomena of interference. Okay, but there are challenges, and challenges are, first of all, you're sampling light by these lenses, and you are limited in terms of resolution. What happens is that if you make these lenses too small, well, you get more sampling of the rays, but you become limited by the point spread function of these lenses, because when you make the lenses small, the numerical aperture suffers. Therefore, you're getting poor quality 2D perspectives of the 3D surface, so your reconstruction suffers. If you make the lenses too big, well, you improve the numerical aperture, but now the problem that you have is that the sampling of the ray has suffered. Field of view, compactness, cost, applications, depth of focus, longitudinal resolution, these are all problems. And again, that's the reason you don't have these commercially available. By the way, so far everything I said was ray optics, but in fact this phenomenon is not ray optics, it's wave optics, because what you have is propagation of the waveforms from the object to the capture and back for the reconstruction. I won't go into the mathematics, but you can use wave optics to describe this phenomenon. Okay, so this very powerful concept for 3D TV, and it's pursued by these electronic giants like Samsung, like Canon, like Sony. They're all looking, Siemens, they're all looking to sell you a 3D TV. So whoever is making your regular TV now is looking at making a 3D TV and selling it to you. The market is really huge. They did some predictions that if you can successfully make a 3D TV, it's like something like a $30-40 billion a year market. So a lot of these companies who have the resources are investing in this. But the principle is simple. 3D object, array of lenses, camera, encode, transmit, use a 2D liquid crystal display or some kind of liquid crystal display, present the perspective images, an array of lenses, and you get the 3D reconstruction. So because it's very nice, there is no laser involved, there's no speckle effects involved. You can use ambient light. So these are some overview papers. If I'm going too fast, if you just search under my name and like Proceedings of the IEEE Journal or Applied Optics or Optics Express, you see papers on the subject showing up. I want to show you the effect of wave optics here. The problem that integral imaging has is because of these lenses, you really have the best image in focus that is in one plane. So anything that's too far away from that plane will appear out of focus. So here's an example of several images in different planes. And this is a geometrical image. This is the point spread function. And this is the diffractive image. You can see that the quality has suffered because now you're going out of focus. And this is when you bring in pixelation. So how do we remedy that? Well, we can remedy that to some degree by what we call this computational optics idea that I had. That is, if you were to make a 3D reconstruction of this scene, where these images are in different planes, you see that the images that are in plane are reconstructed well, but the ones that are out of plane in 3D, they're not reconstructed well. So what do? We can have this principle of aperture coding. That is, now what I have done, I've introduced an amplitude aperture in the plane of these lenses, and I have immediately improved my reconstruction, as you can see here. Of course, this was just amplitude. The best masks are both amplitude and phase, and if you do that, you will improve you will even improve further beyond this. Okay, this is you're looking at the point spread function without the aperture coding, and with aperture coding, you can see that with aperture coding, point spread function meaning that if you have a point source at the input plane of your imager, ideally you would like to reproduce a point source when you reconstruct. It's like a impulse response of the imaging system. So here you're doing fine, here you're not. Why? Because you're now outside of the depth of field of the system, and as a result, your point spread function is not doing well. But with the encoding system that I presented earlier, this has improved. Okay, so here's another example. As you can see, this is 3D image reconstruction before the aperture coding and after the aperture coding. So in conclusion, I think if we use some kind of computational imaging, encoding of the aperture and algorithms, we can improve this. This is a 3D reconstruction of this scene without the computational imaging and with computational imaging. This is pretty decent, as you can see here. It's a little darker than this. Why? Because we have used amplitude modulation. And by the way, nothing is really mechanically moving here. The nice thing about these systems is that once I record the information, because I have the direction and the intensity of light, I can reconstruct the scene from any perspective that I want. Okay, so I can reconstruct from this way, from up close, from far, from different directions. So that's what I'm showing you. I'm showing you this view of the scene from different directions. Now, this is the optics for reconstructing a 3D scene. Okay, if I go back here, you can see that this is my 3D scene. This is a lens array. This is the capture stage, and of course, this is the reconstruction stage becomes the reverse of the capture stage. And what I have done here is that rather than modulating every lens in my system, I've sort of brought this into one plane, and I've done an optical processing to just use one amplitude mask and modulate them. And as you can see that this is better quality than the conventional imaging system. Okay, so here are some more experiments. There's now a lot of interest in viewing 3D with your smartphone, okay? Or I have an iPhone. So here's one experiment with this. This is a Spanish doll. It's a cook. I think it's and this is a paella for those of you who are interested in Spanish cuisine. These are the view images. These are the view images. This is the blow up of the view images. And this is a 3D movie that reconstructs the doll, and you're looking from different perspectives. Now, there's a difference here. This 3D movie is generated when the object is captured optically but is reconstructed by a computer. And this is when the object is captured optically and reconstructed optically. So clearly this is a little better quality here, because when you reconstruct, you are suffering from the numerical aperture of the lenslets. And you might say that if you use a stereo, you get better quality, and you're correct. But remember, here you have no viewing devices, you have full parallax, you have no abnormal effect, you will not have any convergence accommodation conflict. And in fact, you can even use ambient light to do this. Okay, so I have another movie for you. This is done on an iPhone. And an iPhone is very simple. You basically had to, of course, capture these images, process them by some algorithms, and then you display them on an iPhone and simply place an array of lenslets on the iPhone to reconstruct this. So you have full colors. You're using the display resources that are available to you, and as I said, there are no visual fatigue here. What is nice about this, folks, is that if an object is behind occlusion, like trees, bushes, and so on, or a scattering medium, or clouds, or fog, and so on, because you have, as I said, both the intensity and direction of the rays, you can go behind the occlusion and reconstruct this object. You might say, how? Well, the reason being that if you were to do a 2D image of this occlusion and the object will be superimposed because you have only the intensity of the rays. But in this case, because you're getting the direction of the rays as well, you can go behind here and reconstruct this. So this sort of explains what happens here. Objects, occlusion, sensing, okay? And when you reconstruct this, remember, you are following these rays back so you can go behind the occlusion and reconstruct this object, okay? So here you have this car where the letter 3D is obscured. You cannot see it, but in the 3D reconstructed version, you can see the 3D characters 3D. This is a very heavy, this is a scene, 3D scene. This is a very heavy occlusion. As you can see, you can hardly see parts of the scene. This is what we call a true class target. It means that we are looking for this object in this scene, okay? We do not know anything else about these objects. We do not know if this is here or not. We do not know where it is, but with 3D imaging, we were able to pull out this object through this correlation process. This correlation peak tells us that in this scene, there is an object in this location. Okay. Now, things don't have to be in a nice regular structure. Here you have cameras located randomly in space, looking at a scene, and you're able to reconstruct the 3D profile of the scene. Okay, so what is nice here is that because sensors are very inexpensive, so in this room you can place small cameras and then be able to do a 3D reconstruction. So this is very good, I think, for industrial applications where you have to make prototypes of objects very rapidly and make measurements and so on. Okay. This is another work that we are doing. As I said, we're looking at principles of quantum optics here. And I will show you an example. As I said, generally we have plenty of light, plenty of photons. Okay, so for, let's say, 3D imaging, lots of photons coming in. You have the sensor. These are the perspective images. This is with 100,000 photons. And this is the 3D reconstruction of that scene. So I'm doing 3D display, but I have to show you in a 2D viewing format. So I'm showing you one plane at a time. So I've reconstructed 3D scene. I'm showing one plane at a time I'm going through. Okay. So when you reconstruct, for example, this object, of course, these are out of focus. So then when you get to those planes, they're in focus, and so on, so on, so on. But what happens now if I were to substantially reduce my number of photons here? If you come very close to the screen, and you've got my laptop screen, you see this is about 1,000 photons in the scene on the average. So you see some pixels are turned on, and on average about 1,000 pixels get about one photon. It's very unlikely to get more than one photon because the number of photons are so low. But so by looking at that, you don't recognize the scene. However, I can reconstruct this. I wish we could make this darker. No. We still have too many photons here. But it's not bad. And these are not the best algorithms. I can do better than this. But the point of it is that we have been able to do complex things with very few photons. Now, of course, the advantage here is that let's say, if you were to communicate this or store this, you would be sending 1 kilobits rather than many megabytes. Okay. And I look forward, we don't know exactly why this happens. Oh, thank you. We don't know exactly why this happens. I look forward to working with someone to explain to us. But of course, I don't have to tell you this, being in physics, but a single photon, when it's emitted and it's illuminating the scene, it becomes a waveform. So even a single photon has the potential of modulating the entire information, has information about the entire scene, even a single photon, right, as a waveform. But of course, when it's detected, it becomes a particle. And if there was a way that we could retain the information about the scene even in a single photon, I think that would be really, really exciting for the purpose of information processing. Now, here's an experiment where you have this scene in 3D, and again, there's about 1,000 photons used to illuminate the scene. And now this car is the true class target. And you can see that with about 100 photons, you can identify this car, okay, through the process of correlation. So here's the correlation for the true class object, the right car that you're looking for, and the other cars which you don't know anything about. And you can see that with about 100 photons. And this, of course, every time you repeat the experiment, the correlation peak changes because of the Poisson nature of the detection becomes a Poisson process. So every time you repeat the experiment, you don't get exactly the same image. You get a different image. There is a statistical fluctuation. And with 100 photons, you're able to identify the car. Here, we repeated the experiment with this. It's too bad you can't see this because here is an object in a complex background, okay, and these are the photons that are detected from the object without any background or noise. And if you look here, this is the photon. This would be green, this would be red, this would be very noisy. If you could look at my computer screen. And this is the signal plus noise here. There are almost 25 times more noise than there are signals. And even then, of course, now you need more photons. You need maybe hundreds of photons. But even then, we were able to identify this object not using a massive number of photons. And this was done with an optical experiment. We used the cool CCD camera here to get these images. And if you look at my screen here again, this looks like noise. There's a very low photon flux 2D perspective images that are recorded, and we are able to recover this image. This is still raw without additional processing. Can potentially get better than that. I won't explain this to you, but we're using information theory. You use information theory in physics, correct? Yes, no? Okay. So what we're trying to do here is to really use the entropy, which is a measure of information content in a source, is given by this expression, and use the mutual information to see what's the mutual information between the photon counting 3D reconstructed image and the original image. Okay, and if we can optimize this mutual entropy, then of course we can find a way to extract lots of information from the scene with very few photons. So here again are some experiments. A 3D object occluded. This is the normalized mutual information here. And the normalized mutual information is given by the mutual information between the photon counting image and the original image, between basically this scene and that scene, divided by the entropy of the original scene. So it's very interesting here because what you can see is that when you do this in 2D, okay, what happens is that your normalized mutual information is very low, clearly, because you can't really see the object here. But of course, what happens is that your compression is much higher. And when you do this in 3D, you can see that the normalized mutual information is much better because you are able now to visualize the object. Okay. We were asked to see if 3D imaging can be done from behind clouds. They were very interested, for example, to fly over a scene and see if they can look through the clouds on the ground and see what's going on. The way it's done now is through LiDAR. And what they do, they illuminate a laser that goes through the clouds, illuminates the ground, is reflected back or scattered back, and they gate the laser beam to catch the ballistic photons. And what that means is that when the photons propagate through a scattering medium like clouds, very few, go straight through and are bounced back. They don't go through this random walk, okay? The others scatter. And that scattering means that the ones that are scattered, it takes them a longer time to travel from the source, the object, and back to the receiver. So by gating it, as soon as they turn the laser on, the clock starts, and they reject the photons that come back after a certain threshold because they think that they are scattered photons. But they wanted us to do this with basically with passive light, without use of lasers. The problem with lasers is that it's cannot be used on a population, obviously, because of health issues. The system is very bulky, it's expensive, and it's not persistent. You cannot do this all the time. You can do this only for certain periods of time. And so we did some experiments. So we wanted to do this with clouds, but it turns out that if you take water and add milk to it, it has the same properties as clouds. So we took a tank of water, we created this scene, added milk to the water, made it cloudy, and then started doing experiments. So here's a clear scene, clear water, and this is water seen in a milky water or murky water. And I start with a clean. If there is no milk added to the water, you see a nice clean reconstruction of the scene. Okay, you saw the bug, you saw the fish, you saw the shark. One more time. Okay, so the bug shows up first. There's a hidden treasure in here, and that's the shark. With the murky water, you can't see anything because of the scattering. So what we did now, again, going back to computational optics, we took this data and we did a statistical processing on this data. We treated this effect of scattering as noise, and we tried to estimate a signal from noise. So basically, we used a lot of statistics, and this is what we got. It's not bad. Okay, there are some errors here, but overall, pretty good. For the fact that we used basically passive illumination and we have no information about the scene. Okay, this is another topic that we're working on. So far, everything I showed you, the imagers were laterally, the cameras were in XY. So we did some experiments of what happens if you basically approach a 3D object this way. Okay, or you have a stationary camera, a 3D object is approaching it longitudinally. And I want to show you some experiments here. There are a couple of cars occluded here, so you have to pay attention to see them. That's where they are. It's a very substantial amount of occlusion. And again, this is the 3D movie. Okay, they come in and out of focus very quickly. Here it is again, right there. Okay. So that system works too. This is another work that we are doing with microscopy. And the issue, of course, is that when you illuminate a high resolution object, you are limited for practical purposes with the wavelength of light in terms of details of the scene that you can capture. The reason being because of evanescent waves. What occurs is that details that are smaller than wavelength, evanescent waves do not propagate more than maybe a wavelength, and they never reach your sensor. So the objective here is try to catch the evanescent waves. And the way that's done is that if we can modulate, if we can have an encoding pattern very close to the surface of the sample and modulate the evanescent waves before they disappear, then with signal processing, we can recover those. And that's the work of these folks that I'm collaborating with, Ziv Zalewski and his colleagues and Javier Garcia. And I show you. So what did we do? We basically, what we're proposing to do is to float nanoparticles very close to the surface of the sample. Okay? So you can have your sample and think of nanoparticles flowing through like a microfluidic chamber very close to the surface. Now this has to be within a micron of the surface of the sample. So this is what happens. Where are you? Oh, here. So you see that these nanogold particles now are flowing around here, and we make multiple exposures of this. And this multiple exposure allows us to recover the signals like this. So here's the case where this is about 4 micron scale. This is the case where we did not use the suspended nanoparticles. We're using basically gold particles here. And it's the case that we are using them. So clearly we have better resolution here than this case. Yeah. Now, the algorithms we have developed here are simplified. I think that we have to look at the analysis here from a rigorous electromagnetics theory point of view, which we haven't done yet. That's why this result is not as good as it should be. There are simpler algorithms, but the results are not as good. My hypothesis is that because these gold particles are so close to the surface of the cell, you have to use coupled wave theory to analyze this because the boundary conditions change. We are now just using wave optics, diffraction with wave optics. This is another work that I think is interesting. So here, and this work that I mentioned is automated identification of cells by 3D imaging. The idea is that if we use some kind of a microscope, or digital holographic microscope in this case, to reconstruct these cells in 3D, and then inspect these cells, look for various diseases, look for various complications, and so on, we will be able to, interfacing that with a computer, to automatically inspect these and tell you what kind of bacteria it is, or if these are red blood cells, are they healthy red blood cells, or are they infected with some kind of a disease? Okay? And we're looking at trying to do this as simple as possible because we are working with colleagues in India, and the objective is really to take these to the field, have a very simple, robust, compact system that you can take it to the field and try to help people, who maybe are suffering from malaria or suffering from other diseases, to take the information and then maybe interface it with a cell phone, have an app, or send the data to a database somewhere that can come back and say whether someone, what their condition is. These are some experiments we did with stem cells, for example. Stem cells, I don't know if you do stem cell research here in the Czech Republic? There was a time you couldn't do it in the United States, but now you are doing stem cells? Oh, okay. So stem cells, the problem that we were told by our colleagues was that they had a serious problem with cell differentiation. So cell differentiation meaning that you're growing stem cells for a particular specimen. Okay, these stem cells are going to be transplanted for a particular purpose. But sometimes they basically differentiate into a different cell, which can be very serious because if now you put that cell into humans, could be very serious complications. And the way for them to know it was to remove the cell from the culture, stain it, and look at it under a microscope. But by then, the cell is destroyed. So we proposed using our approach, which means that it was non-invasive and it didn't have to be removed, and we could tell them whether there was any cell differentiation or not. So the idea was to illuminate the cells, record the digital holographic microscope images of these cells, reconstruct this in 3D, okay? Go inside the 3D and take samples of this. This is now done in the computer. Take samples of this 3D, generate a statistical population. So now you have a statistical field, and the statistical field can be compared against whatever you wish. If you're looking for a bacteria, if you're looking for a particular disease, you can extract that information during your training stage. And then when a new cell is presented, you go through the same process, obtain the statistical information for the new cell. Now you can do a statistical comparison between your true class hypothesis, as we say in statistics, and the false class hypothesis. These are some microbeads. We are now doing things with microfluidic chambers. So there's a movie of that, and we're looking also temporal information. We're interested in temporal information. This is a red blood cell captured by our digital holographic microscope. So far, I discussed the 3D profile of this, but now we're also interested in what we call motility, that is temporal fluctuations of these cells. And my hypothesis is that if you have a healthy cell versus a diseased cell, this temporal fluctuation could be different. Not only the 3D profile of these cells will be different, just like a healthy person and an unhealthy person. A healthy person moves differently, is more vigorous, and so on. And someone who's ill, probably their temporal movement is different. So I'm showing you now just the temporal fluctuation of this red blood cell in simply one small area. And this is the thickness versus position. This is a cross-section of the RBC. And now this is the temporal fluctuation that you can see that is measured for about 400 seconds. And this is the thickness change across the whole cell between time instance of 75 seconds and 300 seconds. Because what will happen is that the cell is beating, has motility, and if you measure the phase and subtract the phase for that period, then you see this is the phase change due to the temporal fluctuation. This is the Fourier transform of the temporal fluctuation of the cell. So what I really like to do now is to compare this between healthy and diseased cells. And from electrical engineering perspective, there are some very, very good filters that can analyze the Fourier information of signals. So we can apply those tools here and benefit from them. These are some additional experiments of different cells. And this is what we have done. True class cells, false class cells, these are two types of cells. And remember the statistical distribution that I was talking to you about. These are the probability density function of the true class versus the false class, the two different kinds of cells. And as you can see, we can separate them to classify what cells they are. This is what we have put together. This is a digital holographic microscope, and we designed this to be very, very low cost. And the idea is to take this to places like Africa, India, places that are very remote from sophisticated health, medical resources. And this is, for example, is a laser pointer. That is a webcam. This is simply a shearing glass. It's just a glass plate. There is no reference beam here. It's a single beam that goes. It bounces through this glass plate. So the first one goes through, then it bounces, and then it goes through. So that becomes a reference beam. So you can see that it's very compact. This is designed to work even with like a iPhone or smartphone. Okay. This is designed not to have a reference beam. As you know, in holography, you have an object beam and a reference beam. This is really self-contained, so it's very robust, very stable, and very low cost. Okay. These are some results that we have for red blood cells. Okay. We're also looking at cancer cells. This is not a digital holographic microscope. This is an interferometric, is a phase lock interferometer. And what we have is we have a cancer cells that are placed here and an interference path, a very precise interferometer here. And there's phase lock loop here, so there is no need for phase unwrapping. And the stage is scanned, and the phase information of the sample is measured. So the voltage that's applied to this PZT stage compensates the phase change for phase locking. And spatial resolution is less than a micron here. Optical path length, very good, less than 2 nanometers. This was applied to This is a collaboration with Professor Watanabe from Japan, one of my colleagues. So the hypothesis that we have is that cancerous cells, because they are multiplying, they are reproducing uncontrollably, they have to draw more blood. Okay, and when you draw more blood has minerals, has iron, and so on. So when you propagate light through this, it will have a different modulation. So that's our hypothesis, and it turned out to be, apparently for modulating light, turned out to be correct. This is healthy cell versus cancerous cells, and you can see the thickness is different here. Okay, the phase change affects the phase change according to this color encoding here. So we reconstructed these, and we are looking at the probably dense function or the histogram for cancerous cells. This is the optical path difference, by the way, for healthy versus cancerous cells. And you can see that it's different. The cancerous cells have a larger optical path length. And you can, basically you can classify them. This is another kind of 3D microscope based on this principle of integral imaging. So here there is no incoherent light, everything is there is no coherent light, everything is incoherent, and you're looking at the different perspectives of a cell and reconstruct them to do 3D imaging. Problem here is the resolution. You really suffer resolution because of these lenses. But the nice thing is that you can get a 3D profile of the object without doing optical coherence tomography. So in conclusion, I presented you many applications of 3D imaging. Particularly, I showed 3D integral imaging, very attractive for display and computational imaging. We also showed 3D digital holographic microscopy. We also showed applications for low light level imaging. And I believe that you, by using computational imaging, aperture coding, signal processing, information theory, we can address many of the problems that exist in 3D. There are some overview papers. If I went too fast, and then once again, you know, if you Google my name and 3D, you know, different papers, these are overview papers that we can have. Now, I want to ask you, who knows Stéphane de la Roche? Anyone knows here Stéphane de la Roche? From France, not too far from here. In American scales, not too far. Okay, this fellow was incredible, all right. I wish he had lived to be 100 years old. He was a writer, like a science fiction writer, they call it. And he published this stories called Je Fanti. And I think he made a living that way. So every week he would publish a segment of the story, and you had to wait until next week to see what happened. But his story was about a team of French African explorers who discovered the secret land of Je Fanti. Okay, so they traveled to Africa, they found this tribe, and these were like superheroes. Where a race of secret supermen live in royal isolation. The hero, while visiting the dwelling of the tribe chief, notices a window where he observes a seascape, and looking closer, he bumps his head on a device, which I think was the first 3D imager of its time. Now remember, this was published in 1760. 1760 in the United States was the French-American War, the French-Indian War. Photography would be invented almost 100 years later, or maybe 70, 80 years later. Holography would be invented almost 200 years later, okay, just to put things into perspective, all right? TV would have been invented in 180 years from then. All right, now, this is his writing. And I'm putting in red those terms that I think are very important for a good 3D imaging system. Okay. That window, this is what, you remember he bumped his head on a device. He's describing that device in the tribe chief's tent. That window, that vast horizon, those black clouds, that raging sea are all but a picture. The elementary spirits have sought to fix these fleeting images. They have composed a subtle matter by means of which a picture is formed in a twinkling of an eye. Okay, to consider as like photography. They coat a piece of glass with this matter and hold it in front of the objects they wish to paint. The first effect of this canvas is similar to that of a mirror. One sees that all objects near and far, so very high depth of field here. The image of which light can transmit. The canvas, by means of its viscous matter, retains the images. Our canvas shows the objects with the same exactness and retains them all. This impression of the image is instantaneous, so very high speed capture, and the canvas is immediately carried away into some dark place. I guess later on we would call that the dark room for photography. An hour later, the impression is dry, and you have a picture, the more valuable in that it cannot be imitated by art or destroyed by time. The correctness of the drawing, the truth of the expression, the stronger or weaker strokes, the gradation of shades, the rules of perspective, all these we leave to nature, who with a sure and never erring hand draws upon our canvases images which deceive the eye. This is de la Roche in 1760. So I think this guy was a genius. I mean, he predicted it all. You know, he predicted photography, he predicted holography, he predicted integral imaging, the whole thing is there. And with that said, I stop here and thank you for your time.
Now the session is open. Questions from the audience?
Yes.
I would like to ask you one more time about the principle with the array of microlines. I'm not sure
Yes.
in which sequence do you take images from CCD camera. Here is object, here is array, and behind it there is a CCD chip.
Yes.
And you just take picture from the chip, nothing more. And in which sequence or which algorithm you take that?
Okay. So I'm sorry, just to be clear about the question, are you saying if there's a special sequence that we take the pictures?
Yes.
No, they don't have to be. They don't have to be, because basically what you need to have the intensity and the angles. If I go to here, maybe that's a good. Okay, so we need the intensity and the angles. So it really doesn't matter what the sequence is. You can take all the pictures at the same time. You can take them sequentially. You can, if you recall, I showed this example here where, here. This was a random walk. So a camera was moved as a random walk and took pictures. And we were still able to put it together. Because all you need is the intensity and the direction of the rays. Once you have that, you can put it together. If you do things sequentially, then of course it means that your object has to be stationary, that the object is not moving. If the object is moving, you have to record at the same time. So you can have an array of cameras, or you can have cameras located, randomly in space and do this. We even showed that your cameras can be on a non-flat surface. So for example, I can put cameras on a soccer ball, on a football. Okay, you say soccer, you say football. Sorry, on a football, and I can still record and construct a 3D scene. But of course, the math gets more complicated now. You're not on a flat surface; you're on a curved surface, and so it can be very flexible, can be very robust.
Can you tell us some details about the micro lens?
Details about the micro lens.
How do you fabricate them?
Micros. Yeah, micro lenses you can purchase commercially, and they are either refractive, using refractive optics. As you know, you can have a plastic that they stamp and make using refractive optics. You can make them using diffractive optics. You can make them using holography, like form them in some kind of a photopolymer. Basically, you want them to be very good imagers. You want them to have a very good point spread function. That's what you like to have. That's the challenge if...
I was asking about technical parameters in this lens.
Technical parameters of the lens. So can you be more specific, please? Technical parameters? Like how big they are, or what's the focal length? Okay. Yeah, so it depends on your application. For example, if you're doing microscopy, then obviously the size of your micro lenses becomes smaller, right? Because you have a very small scene, and what you like to do is have small micro lenses that sample the rays at a very good sampling rate. So, in that case, usually the diameter of your micro lenses are maybe 100 micron, 50 micron, 100 micron. If you have a cell that's, let's say, 5 microns, 10 microns. For objects that are far, we usually don't use micro lenses. We use array of lenses. And we have multiple cameras because the object is far, and therefore you have to have a good point spread function, numerical aperture, to capture them. And in fact, we did some work where I didn't present it here, but we optimized. We said that, okay, you have an object that's this big and this far. What is the optimum number of lenses? What's the optimum size of the lens? How many pixels? What's the size of the pixels? How many cameras, and so on, so that you achieve certain metrics, like certain resolution, certain depth of field, certain longitudinal resolution, and so on. So that's also possible.
Have you tried to apply this principle to another range of spectrum, for example, infrared?
Yeah, we did. In fact, we did infrared. That was in cooperation with military. We did. I wonder if I can find it here. Yep, here. We did. So that's a mid-wave infrared camera. This is a military base, and this looks out maybe a kilometer away at an object. And the object is behind trees. And we were able to reconstruct. This is the trees reconstructed. This is the object reconstructed. And this is these are buildings that are reconstructed in 3D. So you can get the range as well as the 3D shape of these objects. This was on the Optics and Photonics News. We appeared on the cover for our work on using 3D imaging for recognizing and identifying cells. Yes?
The cancer cell identification is based on optical thickness.
Yes.
Is it relevant to actual hydrogen culture?
Yes, it's the optical path length, yes,
Yeah.
and which depends on the index of refraction.
Yeah.
And
And
so it depends on the thickness of the culture and also the index of refraction. We felt that if the cell is drawing more blood, the index of refraction changes, so the optical phase changes because of that.
yeah, but is there any sort of practical application? Yeah.
I'm sorry?
Any practical application yet done with this approach, or just a kind of pilot study?
It's just a pilot study. I don't think anyone had ever looked at it. So in 3D. So we basically proposed that. We didn't know ourselves first, but it was very interesting that what a difference, it was a very noticeable difference.
So you compare several types of culture.
Oh yes, we had, I didn't give the numbers, but it's in that paper, it's an optics lab in 2013. Yes, we had many samples that we looked at.
And was done on
Not many, I mean by many I meant about 10. It's very hard to get breast cancer samples.
or cultured cells.
Yes.
Yes, and so you put them living into the microscope or
No, I believe they were stained.
stayed.
Yes,
It's not already fixed.
right.
Dehydrated.
I believe so, yes.
Yeah.
Yes.
And what about this?
But in principle, they can be live. They don't have to be.
And the stem cells identification to which part they are going to go.
They were from mice.
Nice.
Yeah, they were.
It was also done on
They were embryonic stem cells from mice.
stained specimen.
I'm sorry.
It was fixed and stained specimen.
No, they were not. They were not stained. No, we looked at it as they were growing. In fact, I can show you
In a chamber.
in the chamber, the regular chamber for growing stem cells.
Chamber for culturing cells.
Yes. Yes, and we looked at it over many days, and we saw the colony grow, and we made measurements of this evolution
So
of the
you
cells.
observed consequences of your prediction.
Yes, we were able to do differentiation
Just watching.
just by making the 3D images and running them through an algorithm for automated identification of differentiation.
So it was confirmed.
Yes, it's powerful. I think it's powerful. Even though it's micro scale, but and the micro range is 3D.
Yeah.
And when we compared 3D with 2D, 3D was a lot better than 2D. 2D was harder to do differentiation than it was in 3D. Yes.
As the thickness of cells is concerned, similar information can be obtained with interference microscopy. Did you perform some analysis concerning the relative sensitivity or?
Right. So I think what you're trying to say is that with holography, we cannot measure the absolute thickness. We get the product of the thickness and the index of refraction. That's correct. That is correct. So, and that's a good point. So everything we have shown here is really the product, the index of refraction, and the optical path. Because to separate the two, then we would need to do something like tomography, where we would have to make many measurements from different angles. And that would be an interesting thing to do. We haven't done it. But for the sake of identification, classification, it didn't seem to be necessary. There was enough information.
It's a little bit different. If you can see some definite advantage of this photographic technique over interference microscopy, which is a standard technique.
Right. I felt that that's a good question. We felt that the system can be very compact, can be very fast, can be single exposure, and the 3D information is very helpful. But I think in principle, they're similar because they're using the principle of interference. It's just that you're talking about some semantic differences, but more or less it's the same principle. It's interference of light.
Another question is completely different, just for curiosity. Is somebody around you, or maybe you, interested in designing techniques or methods that makes it possible to extract a single object plane from a pair of register of error of images?
I'm sorry, can you repeat the question?
You can get the information of an object
Yes.
in a pair of images that are error. And is there some possibility to extract information about some selected object plane from these two? Because if you are watching it through error, you can see the
Right.
distance. Yeah? And
Right.
you can decide this is closer to me, this is farther from me.
Mm-hmm.
And there is a possibility to extract it by
From stereo.
yeah, from stereo.
From stereo. Yeah, from stereo, you would be interpolating. You would be interpolating. It means that if you have a pair of stereo images, okay, and I triangulate so I get the depth of these. I've triangulated. But if I want to look at a different plane, I have to interpolate because I don't have that information. Yeah. It has to be investigated, I think, to see how maybe if there's a lot of correlation between the images.
So, but it's not the object of images.
No, if you're interested, I can think about how it can be done, but I think that the first thing that comes into my mind is that it has to be interpolated because we don't have that plane, that we don't have the rays for that plane. It's different with integral imaging. Integral imaging, you have many, many samples. So, but stereo, you have only two. So it's different.
Okay.
Okay, welcome. Yes, of course.
Through scattering media, through turbid media, and through some obscuration is the same principle of doing that?
Well, the principle is the same, but the algorithms are different. The obscuration from plants and trees distorts light in a different way that turbid water does. Or
Both are based on the new information and all the stuff you need.
they're all based on integral imaging, many views of the scene, and so on. So the sensing is the same in both cases. The processing is different.
I was just interested in enhancing of the PSF, which was not clear to me how
Oh yeah. So the point spread function was enhanced. So what happens is that what we are basically doing by coding the aperture is that we are modifying the diffraction. Okay, so we're modifying the diffraction, and then we do signal processing to recover the information. So that's what we are doing. So you are, in a sense, slowing down the change of light due to diffraction as it propagates through the z-axis. But of course, you're also modifying the diffraction pattern itself. But by doing signal processing, we try to recover it. It's like basically you think of this as annular optics. Think of it as annular optics. What happens is that you have an incoherent imaging system. If you change the aperture, remember that your optical transfer function is basically going to be a correlation of of the modified aperture. So even though you have modified the aperture, you're still generating, through this correlation, an optical transfer function. If you modify it carefully enough that that correlation produces a reasonable optical transfer function, then you have improved your depth of field. Ideally, you want to design this through using a computer and so on, some iterative process that does this well. So everyone here, this is a physics program, right? Is this correct? Is it Department of Physics? Okay, very good. And physics and chemistry, or just physics, physics and mathematics, or only physics? Only physics. Okay, very good. And how many faculty in physics? How many students? How many faculty? No? Small program, big program? 60 students, 60. But you're talking about one class, meaning a graduate class or undergraduate class?
1 year.
In graduate or undergraduate? 60 students undergraduate program. That's very good. And how many graduate students do you know? No?
Depends on the department.
But in physics, physics has different departments?
Yes.
Oh, so it's a big program then. Very good. How many departments in physics? So you have applied physics, you have quantum physics, you have solid state physics, like that? You have different?
About
10 different departments? I see, very good. Mm-hmm. Each department?
Yes.
Very good. Excellent. No, I'm glad to see there's interest in physics. And how many faculty? Nobody knows how many faculty in physics? No? So if you have 10 departments, maybe at least 5 faculty in a department, no? 50 faculty? No? Yeah, our physics department is just a single department. We have probably about 30 faculty in physics. But I'm not in the physics department, I'm in the electrical engineering department. We have also about 30 faculty. So how many of you have been to the United States? No? Just have to come and visit. I guess there is no visa for Czech citizens. It's easy to visit.
There is visa for Czech.
No. Thank you. It was very nice to see you. Yes. Right, because if I don't require a visa, it means that you don't require a visa. It's usually like that.